So hello dear learners, today we are going to cover our unit 5, that is in unit 5 we are going to study chapter 14, that is breathing and exchange of gases. This unit contains 5 chapters, that is second chapter is body fluids and circulation, chapter 16 is excretory products and their elimination, chapter 17 is locomotion and movement, chapter 18 is neutral control and coordination, and chapter 19 is chemical coordination and integration. We are going to study chapter 14. That is in this unit there is given about human physiology. The reductionist approach to study of life forms resulted in an increasing use of physicochemical concepts and techniques. Majority of these studies employed either surviving tissue model or straightaway cell-free systems. An explosion of knowledge resulted in molecular biology. Molecular physiology became almost synonymous with biochemistry and biophysics. However, it is now being increasingly realized that neither a purely organismic approach nor a purely reductionistic molecular approach would reveal the truth about biological processes or living phenomena. Systems biology makes us believe that all living phenomena are emergent properties due to interaction among components of the system under study. Regulatory network of molecules, supramolecules, assemblies, cells, tissues, organisms, and indeed populations and communities each create emergent properties. In the chapters under this unit, major human physiological processes like exchange of gas, blood circulation, locomotion, and movement are described in cellular and molecular terms. The last two chapters point to the coordination and regulation of the body events at the organismic level. Alfonso Corti, Italian anatomist, was born in 1822. Corti began his scientific career studying the cardiovascular systems of reptiles. Later, he turned his attention to the mammalian auditory system. In 1851, he published a paper describing a structure located on the basilar membrane of the cochlea, containing hair cells that convert sound vibration into nerve impulses. The organ of Corti. He died in the year 1888. Chapter 14, that is breathing and exchange of gases. As you have read earlier, oxygen is utilized by the organism to indirectly break down the simple molecule like glucose, amino acid, fatty acids, etc. to derive energy to perform various activities. Carbon dioxide, which is harmful to, is also released during the above catabolic reaction. It is therefore evident that O2 has been continuously provided to the cells and CO2 produced by the cells have to be released out. This process of exchange of O2 from the atmosphere with CO2 produced by the cells is called breathing, commonly known as respiration. Place your hands on your chest. You can feel the chest moving up and down. You know that it is due to breathing. How do we breathe? The respiratory organs and the mechanism of breathing are described in the following section of this chapter. Respiratory organs. Mechanism of breathing vary among different groups of animals depending mainly on their habitats and levels of organization. Lower invertebrates like sponges, coelonean preta, flatworms exchange O2 with CO2 by simply diffusion over their entire body surface. Earthworms use their moist cuticle, and insects have a network of tubes, tracheal tubes to transport atmospheric air within the body. Special vascularized structures called gills, branchial respiration are used by the most of aquatic arthropods and mollusks, whereas vascularized bags called lungs, pulmonary respiration are used by terrestrial forms for the exchange of gases. Among vertebrate fishes use gills, whereas amphibians, reptiles, and birds and mammals respire through lungs. Amphibians like frog can respire through their moist skin, that is cutaneous respiration also. Human respiratory system. We have a pair of external nostril opening out above the upper lips. It leads to the nasal chamber through the nasal passage. The nasal chamber open into a pharynx, a portion of which is the common passage for food and air. The pharynx open through the larynx region into the trachea. Larynx is the cartilaginous box which helps in sound production and hence called sound box or voice box. During swallowing, glottis can be covered by thin elastic cartilaginous flap called epiglottis to prevent the entry of food into the larynx. Trachea is a straight tube extending up to the mid thoracic cavity with divided level of 5 thoracic vertebra into a right and left per primary bronchi. Each bronchi undergoes repeated division to form secondary and tertiary bronchi and bronchioles, ending up in very thin terminal bronchioles. The trachea, primary and secondary and tertiary bronchi and initial bronchioles are supported by incomplete cartilaginous rings. Each terminal bronchiole gives rise to a number of very thin, irregular, walled and vascularized bag-like structure called alveoli. The branching network of bronchi, bronchioles and alveoli comprise the lungs. We have two lungs which are covered by a double-layered pleura with pleural fluid between them. It reduces friction on the lung surface. The other pleural membrane is close contact with the thoracic lining, whereas the inner pleural membrane is in contact with the lung surface. The part starting with the external nostrils up to the terminal bronchioles constitute the conduction part, whereas the alveoli and their ducts form the respiratory exchange part of the respiratory system. The conduction part transports the atmospheric air into the to the alveoli, clears it from foreign particles, humidifies and also brings the air to body temperature. Exchange part is the site of actual diffusion of O2 and CO2 between blood and atmospheric air. The lungs are situated in the thoracic chamber, which is anatomically an airtight chamber. The thoracic chamber is formed dorsally by the vertebral column, ventrally by the sternum, laterally by the ribs, and on the lower side by the dome-shaped diaphragm. The anatomical setup of anatomical setup of lungs in the thorax is such that any change in the volume of thoracic cavity will be reflected in the lung pulmonary cavity. Such an arrangement is essential for breathing, as we cannot directly alter pulmonary volume. Respiration involves the following steps: breathing or pulmonary ventilation, by which atmospheric air is drawn in and CO2 rich alveolar air is reached out. Diffusion of gas O2 and CO2 across alveolar membrane. Transport of gases by the blood. Diffusion of O2 and CO2 between blood and tissues. Utilization of O2 by the cells for catabolic reactions and resultant release of CO2. Cellular respiration as dealt in the chapter 12. Mechanism of breathing. Breathing involves two stages: inspiration, during which atmospheric air is down, and expiration, by which the alveolar air is released out. The movement of air into out and lungs is carried by creating pressure gradient between the lungs and the atmosphere. Inspiration can occur if pressure within the lungs, intrapulmonary pressure, is less than the atmospheric pressure. It means there is a negative pressure in the lungs with respect to atmospheric pressure. Similarly, expiration takes place when the intrapulmonary pressure is higher than the atmospheric pressure. The diaphragm and a specialized set of muscle, external and internal intercostal between the ribs, help in generation of such gradients. Inspiration is initiated by the contraction of diaphragm, with which increase the volume of thoracic chamber in the anteroposterior axis. The contraction of external intercostal muscles lift up the ribs and the sternum, causing an increase in the volume of thoracic chamber in the dorsoventral axis. The overall increase in the thoracic volume causes a similar increase in pulmonary volume. An increase in pulmonary volume decreases the intrapulmonary pressure to less than the atmospheric pressure, which forces the air from outside to move into the lungs. It means inspiration, relaxation of the diaphragm and the intercostal Muscle return diaphragm and sternum to their normal position and reduce the thoracic volume and thereby the pulmonary volume. This leads to an increase in intrapulmonary pressure to slightly above the atmospheric pressure, causing the expulsion of air from the lungs. It means expiration. We have the ability to increase the strength of inspiration and expiration with the help of additional muscle in the abdomen. On an average, a healthy human breathes 12 to 16 times per minute. The volume of air involved in the breathing movement can be estimated by using a spirometer, which helps in clinical assessment of pulmonary functions. Respiratory volumes and capacities. First is tidal volume, inspiratory reserve volume, expiratory reserve volume, residual volume, expiratory capacity, functional residual capacity, vital capacity, total lung capacity. Tidal volume of air inspired or expired during a normal respiration. It is approx 500 ml. It means a healthy man can inspire for only 6000 to 800 ml of air per minute. Inspiratory reserve volume, IRV. A person can inspire by a forcible inspiration. This averages to have 2500 ml to 3000 ml. Expiratory reserve volume, a person can expire by a forcible expiration. This averages 1000 ml to 1100 ml. Residual volume of air remaining in the lungs even after the forcible expiration. This averages 1100 ml to 1200 ml. By adding up of air respiratory volume described above, one can derive various pulmonary capacities which can be used in clinical diagnosis. Inspiratory capacity, total volume of air a person can inspire after a normal expiration, and this includes tidal volume and respiratory reserve volume, TV plus IRV. Expiratory capacity, total volume of air a person can expire after normal inspiration. This includes tidal volume and expiratory reserve volume, that is TV plus ERV. Functional residual capacity, volume of air that will remain in the lungs after a normal expiration. This includes ERV plus RV. Vital capacity, the maximum volume of air a person can breathe in after a forcible expiration. This includes ERV, TV, IRV, or the maximum volume of air a person can breathe out after a forced inspiration. Total lung capacity, total volume of air accommodated in the lungs at end of forced inspiration. This includes RV, ERV, TV, and IRV, or vital capacity plus residual volume. Exchange of gases. Alveoli are the primary sites of exchange of gases. Exchange of gases also occur between blood and CO2, and CO2 are exchanged in these states by simple diffusion. Simple diffusion mainly based on pressure or concentration gradient. Solubility of the gases as well as thickness of the membranes involved in diffusion are also some important factors that can affect the rate of diffusion. Pressure contributed to an individual gas in the mixture of gases called partial pressure and is represented by PO2 for oxygen and PCO2 for carbon dioxide. Partial pressure of these two gases in the atmospheric air and the two sides of diffusion are given in table 14.1. Atmospheric air is 159 in millimeter Hg, 0.3 of CO2, alveoli 104, 40, blood that is deoxygenated 40 and 45, blood that is oxygenated 95 and 40, tissues 40 and 45, O2 and CO2 in mmHg. Similarly, a gradient pressure for CO2 in the opposite direction. It means from tissues to blood and blood to alveoli, as the solubility of CO2 is 20-25 times higher than of O2. The amount of CO2 that can diffuse through the diffusion membrane per unit difference in partial pressure is much higher compared to that of O2. The diffusion membrane is made up of three major layers. Figure 14.4. These are layers. This is a blood capillary, blood is going, and this is one cell thick. These are one cells. squamous epithelium and of alveolar wall. This is alveolar cavity, and this is blood capillary. This is yellow substance is basement substance, namely a thin squamous epithelium of alveoli and the endothelium of alveolar endothelium of blood capillary. And here is given endothelium of alveolar capillary or blood capillary. And the basement substance. This yellow part is basement substance, composed of thin basement membrane supporting the squamous epithelium and basement membrane surrounding a single layer of endothelial cells of capillaries in between them. However, its total thickness is much less than a millimeter. Therefore, all the factors of our body are favorable for diffusion of CO2 from alveoli to tissues and that of CO2 from tissues to alveoli. Transport of gases. Blood is a medium for transport of O2 and CO2. About 97% of O2 is transported by RBCs in the blood. The remaining 3% of the O2 is carried in a dissolved state through the plasma. Nearly 20-25% of CO2 is transported by RBCs, whereas 70% of it is carried by bicarbonate. About 7% of CO2 is carried in a dissolved state through plasma. Transport of oxygen. Hemoglobin is a red colored iron containing pigment present in the RBCs. O2 can bind with hemoglobin in a reversible manner to form oxyhemoglobin. Each hemoglobin molecule can carry a maximum of 4 molecules of O2. Binding of oxygen with the hemoglobin is primarily related to partial pressure of O2, partial pressure of CO2, hydrogen ion concentration and temperature are the other factors which can interfere with the binding. Sigmoid curve is obtained when percentage saturation of hemoglobin with O2 is plotted against the PO2. This one is called oxygen dissociation curve. This curve is called oxygen dissociation curve. Oxygen dissociation curve. This diagram and is highly useful in studying the effect of factor like PCO2, H plus concentration, etc. on binding of O2 with hemoglobin in the alveoli, where there is a high PO2, low PCO2, lesser H plus concentration and lower temperature. The factors are all favorable for the formation of oxyhemoglobin. Whereas in the tissues where low PO2, high PCO2 and high H plus concentration and higher temperature exist, the conditions are favorable for dissociation for oxygen from the oxyhemoglobin. This clearly indicates that O2 gets bound to hemoglobin in the lung surface and gets dissociated at the tissues. Every 100 ml of oxygenated blood can deliver around 5 ml of O2 to the tissues under normal physiological conditions. Transport of carbon dioxide. CO2 is carried by hemoglobin and carbaminohemoglobin. About 20-25% is binding related to the partial pressure of CO2. PO2 is major factor which could affect this binding. When PCO2 is high and PO2 is low, as in tissues, more binding of carbon dioxide occurs. Whereas when the PCO2 is low and PO2 is high, the alveoli dissociation from CO2 from carbaminohemoglobin takes place. It means CO2 which is bound to hemoglobin from the tissues is delivered at alveoli. RBCs contain a very high concentration of the enzyme carbonic anhydrase in minor quantities of the same present in the plasma too. This enzyme facilitates the following reaction in both directions. CO2 plus H2O carbonic anhydrase. It is a reverse reaction. H2CO3 also a reverse reaction as CO3 minus plus H plus. At the tissue site where the partial pressure of CO2 is high due to catabolism, CO2 diffuses into blood, RBCs and plasma and forms HCO3 minus and H plus. At the alveolar site where PCO2 is low, the reaction proceeds in the opposite direction, leading to the formation of CO2 and H2O. The CO2 trapped as bicarbonate at the tissue level and transported to the alveoli is released out as CO2. Every 100 ml of deoxygenated blood delivers approximately 4 ml of CO2 to the alveoli. Regulation of respiration. Human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demands of body tissues. This is done by the neutral system, a specialized center present in the medulla region of the brain called respiratory rhythm. Center is primarily responsible for this regulation. Another center present in the pons region of the brain called pneumotaxic center can moderate the functions of the respiratory rhythm center. Neural signal from this center can reduce the duration of respiration and thereby alter the respiratory rate. A chemosensitive area is situated adjacent to the rhythm center, which is highly sensitive to CO2 and hydrogen ions. Increase in these substances can activate this center, which in turn can signal the rhythm center to make necessary adjustments in the respiratory process by which these substances can be eliminated. Receptors associated with aortic arc and carotid artery can also can recognize changes in CO2 and H plus concentration and send necessary signals to the rhythm center for the remedial action. The role of oxygen in the regulation of respiratory rhythm is quite insignificant. Disorder of respiratory system. Asthma is a difficulty in breathing causing wheezing due to inflammation of bronchi and bronchioles. Emphysema is a chronic disorder in which alveolar walls are damaged, due to which respiratory surface is decreased. One of the major causes of this is cigarette smoking. Occupational respiratory disorders in certain industries, especially those involving grinding or stone breaking, so much dust is produced that the defense mechanism of the body cannot fully cope with the situation. Long exposure can give rise to inflammation leading to fibrosis, proliferation of fibrous tissues, and thus causing a serious lung damage. Workers in such industries should wear protective masks. Summary: cells utilize oxygen for metabolism and produce energy along with substances like carbon dioxide, which is harmful. Animals have evolved different mechanisms for transport of oxygen of the cells to the cells and for the removal of carbon dioxide from there. We have a well-developed respiratory system comprising two lungs and associated air passages to perform this function. The first step is for respiration is breathing, by which atmospheric air is taken in inspiration and the alveolar air is released out. Expiration: exchange of O2 and CO2 between deoxygenated blood and alveoli. Transport of these gases throughout the body by blood. Exchange of O2 and CO2 between the oxygenated blood and tissues, and utilization of O2 by the cellular respiration are the other steps involved. Inspiration and expiration are carried out by creating pressure gradients between the atmosphere and the alveoli with the help of specialized muscles, intercostals, and diaphragm. Volumes of air involved in these activities can be estimated with the help of a spirometer and are of clinical significance. Exchange of O2 and CO2 at the alveoli and tissue occur by diffusion. Rate of diffusion is dependent on the partial pressure gradients of O2, PO2 and CO2, PCO2, their solubility as well as the thickness of diffusion surfaces. These factors in our body facilitate diffusion of O2 from the alveoli to the deoxygenated blood as well as from oxygenated blood to the tissues. The factors are favorable for the diffusion of CO2 in the opposite direction. It means from tissues to alveoli. Oxygen is transported mainly as oxyhemoglobin in the alveoli where PO2 is high. O2 gets bound to hemoglobin, which is easily dissociated at the tissue where PO2 is low and PCO2 and H plus concentration are high. Nearly 70% of carbon dioxide is transported as bicarbonate HCO3 minus with the help of the enzyme carbonic anhydrase. 20 to 25% of carbon dioxide is carried by hemoglobin as carbaminohemoglobin. In the tissues where PCO2 is high, it gets bound to blood, whereas in the alveoli where PCO2 is low and PO2 is high, it gets removed from the blood. Respiratory rhythm is maintained by the respiratory center in the medulla region of brain, pneumotaxic center in the pons region of the brain, and the chemosensitive area in the medulla can alter respiratory mechanism. So our chapter is finished. Thanks for watching.